Good day, everyone. Today we will be just talking about biochemical energy production. So I really wanted to do this lecture face-to-face kaso due to the inclement weather, na-cancel din yung synchronous classes natin. So I hope this video recording will be enough. Okay? So here are the objectives for today's lecture. We want to have an introduction to metabolism and the important nucleotides involved in the metabolic processes in our body. We want to discuss how the body produces biochemical energy, how the products of metabolism of your digestion and the various processes in your body enter the Krebs cycle or yung tinatawag nating citric acid cycle, and how that cycle leads to production of other intermediates and energy itself. And as well as we want to understand the mechanism of how ATP is formed by the electron transfer chain found in the mitochondria via oxidative phosphorylation. So what is metabolism? First and foremost, metabolism is the sum total of all biochemical reactions that take place in a living organism. There are two types: catabolism, in which your large molecules are broken down into smaller pieces, and anabolism, wherein small molecules are joined together in order to form larger ones. So catabolism is breakdown. So tandaan nyo na lang, yung mga cat, mahilig silang manira ng gamit. So you break down molecules. That's catabolism. If you build up molecules, or for example, you have two, and then you have monomers, you put them together into polymers, that's anabolism, where small molecules are joined together in order to form larger ones. Okay? So, sa metabolism ng katawan natin, meron tayong mga tinatawag uh, before I forget to mention, your catabolism, your process of breaking down larger molecules into smaller ones, usually ends up producing energy. Kasi yung molecules na yun, meron silang inert potential energy in them. And when you break that down, the energy is released. Meanwhile, if you want to put together different substances, when you want to join them together, you usually consume energy. Kasi you're trying to put these things together. And that requires force. Okay? So next, yung mga substances in our body, the molecules, they usually go through uh, so they can go through a series of processes that we call a metabolic pathway. So metabolic pathway is a series of consecutive biochemical reactions used to convert a starting material into an end product. So for example, meron kang glucose that you got from the food that you ate. That glucose will undergo through what you call the glycolysis pathway, wherein you break down your glucose into the end product, which is pyruvate. So then pyruvate can be converted into acetyl-CoA, then enter another metabolic pathway. So there are two types of pathways, of course. Meron tayong linear, which is isang direction lang siya. And then you have your cyclic pathway. So pag cyclic, syempre, para siyang linear, pero the end product is actually the starting substance sa simula pa din. So you regenerate the starting product if you have a cyclic metabolic pathway. So an example of a linear metabolic pathway that we will be discussing in the future, of course, I mentioned earlier, is the production of pyruvate from glucose. So that's what happens when you break down glucose into break down glucose. Okay. So, and then an example of cyclic metabolic pathway, which we will encounter later on in this lecture, is the Krebs cycle, which is a very important pathway that occurs in our body. Okay? So, before we go right into the pathway, I want to mention several important nucleotides that we will be encountering throughout the course of this lecture. So, I want to give a short introduction to these nucleotides because they have several key functions that occur repeatedly in metabolic pathways that we will consider. So, if we know them, mas madali nating maintindihan yung pathways na i-discuss natin mamaya. Okay? So, the first one, of course, is your adenosine phosphates. So, dito nagbe-belong yung ating ATP. Your adenosine phosphates are made up are adenosine nucleosides. So, di ba pag nucleosides natin is meron kang nitrogenous base at meron kang ribose. Ribose siya kasi adenosine, di ba? Hindi siya deoxyadenosine. So, ribose siya. So, it's an adenosine plus a nitrogenous base nung meron siyang ring dyan. So, do you know this is this is an adenosine nucleoside with a phosphate group, phosphate group, phosphate with a phosphate group or phosphate groups attached. So, pwedeng isa, pwedeng dalawa, pwedeng tatlo. So, pag isa lang, it's monophosphate. Pag dalawa, it's diphosphate. Pag tatlo, it's triphosphate. So, ito nasabi na rin naman natin to several times before that uh, when you lyse a bond between the phosphates, you produce energy. So, what are the bonds pala in your adenosine, diba? So, the first bond, of course, is the phosphoester bond. Yung phosphoester, say phosphoester man siya, is the bond between the first phosphate and the ribose molecule. So the phosphate and ribose, that's a phosphoester bond. While you call the bonds between your phosphates as phosphoanhydride bonds. Bakit siya naging anhydride bonds? Kasi nagtanggal ka ng water in order to form this bond. So anhydride, so remove a water in order to connect these three phosphates together. So these phosphoanhydride bonds are actually very strained or are very actually reactive or strained bonds that require less energy to break. And when you break them, the tendency when you break them, you usually use water. Kaya siya so your these adenosine phosphates, your ATP and the ADP in particular, readily undergo hydrolysis, which releases energy, hydrogen ions, and inorganic phosphate. So, yun yung tatlong products ng, yun ang three of the common products ng hydrolysis ng any of these adenosine phosphates, no? So you have a phosphate, a hydrogen ion, energy, and of course the adenosine phosphate na -1 phosphate siya. So ang nangyayari when you break these bonds, you release energy. Kaya nga si ATP natin is the energy currency of the cell. So when you lyse your ATP, you form ADP and you release energy. Okay? So, ganun yung nangyayari sa ating adenosine phosphates. So, actually, yung ATP is only the most prevalent nucleotide triphosphate. Meron din tayong other nucleotide triphosphates that we will be encountering, and they also similarly contain a lot of energy. So, an example of which is guanosine triphosphate, which we will encounter in the Krebs cycle. And then you also have uridine triphosphate as well as cytidine triphosphate. So uridine triphosphate is involved in carbohydrate metabolism, while cytidine triphosphate is involved in lipid metabolism. But of course, the most common is still your adenosine triphosphate. Okay? Next. Let's go to your next topic with next nucleotide of note is which is your flavine adenine dinucleotide. So bakit siya naging flavine adenine dinucleotide? And what is it? So flavine adenine dinucleotide or si FAD is a coenzyme involved in redox reactions. So redox reactions, of course, alam niyo na to from your chemistry. This is for your oxidation reduction reaction. So pag orgchem, when we're talking about oxidation, there is hydrogen atom loss. When we're talking about reduction, there is hydrogen atom gain. So itong flavine adenine dinucleotide, bakit siya tinawag na ganito? Is because it contains a flavine, it contains a ribitol, and an adenosine diphosphate. So bakit siya naging dinucleotide? Because your ribitol is a reduced form of your ribose. So para siya naging dinucleotide. So flavine adenine dinucleotide. So and it's derived from vitamin B2, which is riboflavin. So, ribitol plus flavin, it's riboflavin. So, kaya siya naging flavine adenine dinucleotide. Okay? So, there are two forms of your flavine adenine dinucleotide kasi nga involved siya in redox reactions. It can serve as an oxidizing agent, no? So, when your FAD is oxidized, in oxidized form, you refer to it as FAD. When you reduce that FAD, it becomes FADH2. So, where do you usually find these FAD molecules? They typically serve as oxidizing agent in a reaction with single bonds, no? Between two carbons to produce a double bond between two carbons. So, makikita natin to later on sa mga reactions na kung ano yung ginagawa ng FAD, no? So, we will go back to this later on para makita nyo yung example niya. So, ano nangyayari dito? Your FAD, when it's oxidized, di ba sabi natin, when it's reduced rather, When it's reduced, there is hydrogen atom gain. So, nagkaroon siya ng additional H dito and then another additional H dito. So, nagkaroon ka dito ng FAD-H2. Okay? So, oxidized form is FAD. Reduced form is FAD-H2. Tandaan yan. Okay? Next. Next naman, we have another coenzyme called nicotinamide adenine dinucleotide. So this one is also a coenzyme involved in redox reactions, but this one is derived instead from vitamin B3. Vitamin B3 is also known as niacinamide, niacin, or nicotinamide. So dun siya nanggaling. So recall ulit natin, redox reactions we're talking about. Uh, uh, kapag organic chemistry in redox, we're talking about hydrogen atom loss if it's oxidation. Reduction is hydrogen atom gain. So yung oxidized form ni NAD is NAD plus, while the reduced form is NADH. So tandaan nyo, pag may H, yun yung reduced form. So parang kanina, di ba? Si FAD and then si FAD-H2. Dito si nicotinamide, si NAD, this one is NADH. So ito namang si NAD serves as a typically serves redox reactions wherein it serves as an oxidizing agent in the oxidation of a secondary alcohol in order to produce a ketone. So kung OH yan ganyan, gagawin niyang, kunyari ganyan siya. Oh, sorry. Kung OH yan ganyan, magiging si double bond O. Gets? So, magiging, imbes na OH siya, magiging, imbes na alcohol, magiging siyang ketone. So, makita rin natin example nitong NADH2. NADH na to, kung saan siya nakatagpuan sa cycle natin, sa Krebs cycle. Okay? So, next. Dito naman tayo sa third, I think this is the fourth important nucleotide that we'll be discussing. So, this one is coenzyme A. So, itong si coenzyme A, you usually refer to it as CoA or co-wash kasi meron siyang thiol group. So, tandaan nyo, si CoA has a thiol group. Okay? So, this coenzyme A is a coenzyme that serves as an acetyl group carrier. So, ano yung acetyl group? Meron kang dalawang carbons, tapos meron kang si double bond O. So, yun si acetyl group. So, yung acetyl group na yun, yun ang kinikerry ng ating, yun yung kinikerry ni coenzyme A. So, if you're talking about coenzyme A lang, ito lang yun. Okay? Itong CoA, SH. Kaya siya naging co-wash. So, itong si CoA plus SH, yan si coenzyme A. Pag nag-carry na siya ng acetyl group, magiging acetyl CoA. Okay? And then, this acetyl CoA is the starting substance sa ating citric acid cycle. So, magitan natin yan, papasok yan sa citric acid cycle. And we will see how everything starts from there. Okay? So, the active portion of this coenzyme A is actually the SH group. So when it carries an acetyl group, it forms a thioester one. Diba sabi na nga natin, your acetyl-CoA is a thioester. Sinabi na natin yan during sa the functional groups lecture. So, tandaan nyo. So, the acetyl group bonds to co-wash through the thioester bond to form your acetyl-CoA. So, this is the whole formula of your acetyl-CoA. So, ito lang actually yung end na may SH yan. So, dyan nagbabind yung itong si acetyl group. Kaya siya nagiging acetyl-CoA. Okay? So next, I forgot to mention that your coenzyme A is derived from vitamin B5. So vitamin B5 is pantothenic acid. So tatlong na yun nabanggit natin B vitamins ngayon. No? So your NAD is derived from riboflavin, si vitamin B2. Your NAD is derived from vitamin B3, which is derived from niacin, vitamin B3, which is your niacin or nicotinamide, and then your vitamin B5, your pantothenic acid, which gives rise to your coenzyme A. Okay? So now let's go to the bulk of the discussion, which is the stages of biochemical energy production. So first, let's go have an overview onto the steps of biochemical energy production before we go right into the Krebs cycle. Okay? So during the biochemical energy production, of course, nagsisimula yun lahat sa pag-digest ng kinakain nating pagkain. So, of course, when you eat something, uh, for example, you ate rice, the rice you eat will go into your mouth and it will become digested primarily using your salivary amylase. So, yung mga proteins mo usually hindi pa siya dinadigest doon. Rather, it will be digested once the food bolus comes into the stomach. So, pagpasok niyo sa stomach mo, mag-act na yung gastric juice, mag-act na yung pepsin. Your digestive enzymes in the stomach will primarily digest proteins. And once these substances pass the stomach and go through the small intestine, doon nangyayari yung majority of the digestion as well as yung emulsification of fats natin using your bile salts. So after digestion, majority of the digestion actually happens in the small intestine. After ma-digest na siya fully, the food that we ate will get absorbed through the intestinal membranes and into the blood. So once it's in the blood, of course, it can get distributed. The various list of substances can be distributed throughout the body na to wherever the body needs these materials. Okay? So, these products of your digestion eventually go into step 2. No? So, the step 2 is acetyl group formation. So, in step 2, the small molecules from digestion are oxidized into acetyl units. So, acetyl units, so yung sinabi natin kanina, na meron kang dalawang carbon, so that's an acetyl, di ba? So, acet, kasi parang F. So, dalawang carbon, may C double bond O, tapos may isang CH3 sa dulo. So, ganun yung acetyl. So this acetyl group conformation may occur in the cytosol or mitochondria. So, the primary products of acetyl group digestion are your acetyl units attached to coenzyme A as acetyl-CoA. So, and as well as the reduced coenzyme NADH. So, so after these two processes, diba, nag-digest ka, you oxidize the products of digestion, you form acetyl-CoA, then your acetyl-CoA will enter now into the third part of the stage, into the third stage, which is your citric acid or your Krebs cycle. So, this Krebs cycle actually occurs in the mitochondria, particularly the matrix. So here in the Krebs cycle, yung acetyl-CoA mo will be oxidized in order to produce energy. So the energy may be lost as heat or may be carried by reduced coenzymes NADH and PADH into the next stage, which is the one we will mention after this. So your acetyl groups can also be oxidized into the carbon dioxide. And this carbon dioxide that you produce during the citric acid cycle is actually the one that you exhale as part of your breathing process. So inhale, and then exhale, di ba? Sabi natin, so inhale, you take in oxygen, exhale, you take out the waste products of your respiration. So that's CO2 from the Krebs cycle, no? So, doon ang gagaling yung CO2, hindi siya nanggaling from nowhere, no? So, it's all coming together. So, next, the fourth stage naman, as we mentioned, is your electron transport chain and oxidative phosphorylation. So, I'm sure I've already shown you the diagram nung sa mitochondria, diba? After you digest your food, papasok siya sa after your glucose has been broken down into pyruvate, your pyruvate converted to acetyl-CoA, then your acetyl-CoA will in the mitochondria, enter the mitochondria, and then this acetyl-CoA will enter the Krebs cycle. And then the products of the Krebs cycle can produce, like your NADH and your FADH2 can enter the electron transport chain and there will be oxidative phosphorylation. So your ETC and your citric acid cycle both occur in the mitochondria. So the NADH and FADH2, which are products of the Krebs cycle, will supply hydrogen ions and electrons needed for production of ATP. So dito ginagamit yung molecular oxygen that we use in breathing in order to for this electron transport chain to occur. So, yung inhalation mo, so pag nag-inhale ka, that oxygen, you use it in the electron transport chain. When you exhale, the carbon dioxide that comes out is from the Krebs cycle. So, when you inhale, it's for the ETC. When you exhale, it's from the Krebs cycle. Okay? So, here is a short summary lang of what happens during the 4 stages of biochemical production. So, stage 1, there is digestion, wherein large complex molecules from your food are broken down into smaller, simpler ones and absorbed through your intestines. When these products are degraded into smaller units and carried throughout the body, uh, it will become even degraded even more into two carbon acetyl groups that become part of your acetyl-CoA. Your acetyl-CoA is then oxidized to produce carbon dioxide, NADH, and FADH2 in the citric acid cycle once it enters the cycle, no? And then this FADH2 and NADH will enter the electron transport chain in order to power the in order to provide the electrons needed to power the proton pumps in the electron transport chain to produce your ATP. So, we will go through that I'm sure medyo nakakalula pa yung mga pinagsasabi ko. So, we will go through that very slowly so that you will be able to understand this topic. Okay? Okay? So, next. So, let's go to your citric acid cycle or the Krebs cycle. So, isa lang si citric, I'm sure nasabi ko na to during the introduction to the lecture, to the cell, no, during the mitochondria. And I'm sure you've already seen this image of the mitochondria. So, tinanggal ko lang yung blow apart dito so that you can see it closer. So, di ba sabi natin, when you eat rice, you produce glucose. When you break down the rice, you produce glucose. The end product of glucose metabolism pyruvate will enter the mitochondria and produce your acetyl-CoA. So, ito yung second step dun sa second stage of biochemical energy production, di ba? Your smaller products are broken down into even smaller two carbon units, your acetyl units, and then at that acetyl unit will combine with your co-wash. So, acetyl unit plus co-wash, you form your acetyl-CoA, and that acetyl-CoA will enter the citric acid cycle. So, then after the citric acid cycle, some of the products of that cycle will carry electrons into the electron transport chain and power the production of ATP. So, so ito muna yung focus natin, no? Kasi itong part na to, we will discuss this during the finals as well as yung other pathways that we see on this picture. So, dito muna tayo sa citric acid cycle natin, and then later on in this lecture, we will focus on the electron transport chain. So, what is the citric acid cycle? So, your citric acid cycle is also known as your tricarboxylic acid cycle. So, it's also known as the Krebs cycle, as the entire pathway was postulated by Hans Adolf Krebs, which allowed him to win the Nobel Prize in medicine in 1953 because he was able to put the whole cycle together. But actually, the person who actually came up with majority of the reactions is actually this guy, si Albert St. Georgi. But the one who put it together was Hans Adolf Krebs. So, hindi pa complete picture uh, yung kay Albert St. Georgi. Tapos, yung na-complete na siya, si Krebs yung nag-complete. So, si Krebs yung nanalo ng Nobel Prize in medicine. So, some people also call this the St. Georgi Krebs cycle. Pero kasi, syempre, ang haba nun. So, usually, yung Krebs cycle yung nagsistick since he was the one credited majorly for it anyway during the Nobel Prize, diba? So, your citric acid cycle is also known as the tricarboxylic acid cycle. Bakit? At bakit din siya tinawag na citric acid cycle? Kasi during the first step of your citric acid cycle, you produce citric acid. And the citric acid has the following formula. So ito yung formula ng citric acid mo. Meron siyang COOH, meron siyang isa pang COOH, and meron pa siyang isa pang COOH. So tatlo yung carboxylic acid niya, kaya siya tinawag na tricarboxylic acid ni Hans Adolf Krebs. Pero hindi kasi siya sure kung citrate nga ba yun. Yung nakita, na-isolate niyang tricarboxylic acid. So, kaya ang unang tinawag niya muna, tricarboxylic acid cycles. Pero na-confirm nila later on that the tricarboxylic acid he was talking about was actually citric acid. That's why it eventually became named the citric acid cycle. So, your citric acid cycle is a series of biochemical reactions in which the acetyl portion of acetyl-CoA is oxidized into carbon dioxide, FADH2, and NADH. Okay? So, it's oxidized into CO2 rather, and then reduced coenzymes FADH2 and NADH are produced. So, let's edit this one lang para hindi na natin siya problemahin, no? Wait lang. Okay. So, ayan. So, this is a series of biochemical reactions in which the acetyl portion of acetyl-CoA is oxidized to uh, CO2 and reduced coenzymes FADH2 and NADH are produced. So, this reaction actually happens in the mitochondrial matrix except for one reaction. So, one reaction actually occurs in the inner mitochondrial membrane because the enzyme is attached to the inner mitochondrial membrane. So all of them except for one will occurs inside the matrix. Okay. So four of the reactions in the citric acid cycle are carboxylation reactions, while two of them are decarboxylation reactions. So we will go into that later on. Kung which reactions actually occur as oxidation, which reactions are decarboxylation. Since we will go through the cycle step by step. Okay. So let's go to the first step of your Krebs cycle. So your Krebs cycle starts with the formation of your citrate. So citrate is citric acid, tapos pag tinanggal mo yung mga H doon sa OH niya. So that's citrate. So, di ba pinakita ko sa inyo yung formula ng citric acid kanina. So this one is citric acid, COH, COH, COH. So pag tinanggal mo yung mga H doon sa dulo, that you form your citrate. So, in forming citrate, the reaction is actually called condensation. Kasi you condense two things together in order to form something new. So, and the enzyme responsible for that is your citrate synthase. Kasi you are synthesizing citrate. So, citrate synthase, what are the raw materials that citrate synthase uses? It uses oxaloacetate and acetyl-CoA. So, acetyl-CoA, as mentioned, it has 2 carbons. And then your oxaloacetate has 4 carbons. So, di ba? So, when you put them together, a 2 carbon plus a 4 carbon, you form a 6 carbon molecule. So, pag pinagsama mo silang dalawa, dito, si acetyl-CoA, you add it here, you form citryl-CoA. And when you remove this CoA part, using citrate synthase, you hydrolyze this bond. So, you put water and then you remove that bond. You've come up with citrate, no? So, si citrate yung magiging product niya. And then, CoA plus a hydrogen ion. So, ito yung nangyayari sa formation of your citrate. So, step 1, you form citrate from oxaloacetate plus acetyl-CoA. Okay? Ulitin natin para mas malinaw. So, in the step 1, the process is condensation. You condense oxaloacetate and acetyl-CoA in order to form your citrate. And the enzyme responsible is your citrate synthase. Yun lang yun. Okay? Next. In the second step of the Krebs cycle, syempre dito ka na mag-start din sa citrate, no? So, in the second step, your citrate becomes isomerized. So, ano yung isomerization? Sinabi natin, di ba? You have the same molecule, but you form an isomer. So, you rearrange the molecule. Same pa rin yung mga components niya, but you just rearrange it. How does this reaction do that? It uses the enzyme aconitase. So, aconitase, catalyze a dehydration-hydration reaction in order to transfer the OH group on this carbon into the other carbon. So, yun lang ang nangyari sa pag-dehydrate niya, no? So, tinanggal niya lang yung OH dito para ikabit niya ng magkabaliktad dito sa kabilang side. So, from citrate, you form your isocitrate. So, tandaan, step 1, step 1, you have oxaloacetate plus acetyl-CoA to form your citrate in step using citrate synthase. And then in step 2, you have citrate isomerization and converting your citrate into isocitrate. Okay? So next step is the oxidation of your isocitrate and the formation of one of the moles of carbon dioxide. So in this reaction, the enzyme responsible is your isocitrate dehydrogenase. So isocitrate dehydrogenase is referred to as the rate-limiting enzyme of your Krebs cycle because it serves as a control point as well as it's the slowest reaction in the entire cycle, no? So, ano nangyayari during oxidation of isocitrate? So, first off, there is uh, the OH group in your isocitrate is oxidized to a ketone by a NAD. So, di ba sabi natin, your NAD usually serves as an oxidizing agent in order to convert an alcohol into a ketone. So, ganun yung nangyari dito, di ba? You have an alcohol here in this carbon, and then you convert it into a ketone. So, your NAD serves as the oxidizing agent. Gets? So, yung H niya, mapupunta kay NAD, magiging NADH and H+. Kasi meron ka pang isang H dito. So, magiging NADH plus H plus siya. So, OH in high societrate is oxidized to form a ketone by NAD. So, the H plus and the two electrons are transferred to NAD from NAD plus to NADH, and then extra H plus is released. Okay? So, pag naging C double bond O na yung carbon dito instead of having an alcohol, you call that substance now as oxalosuccinate. And then your oxalosuccinate is decarboxylated. So, ano ibig sabihin ng decarboxylated? You remove a carbon dioxide. So, when you decarboxylate, you remove the carboxyl, you remove the CO2, then you give rise to a molecule called alpha ketoglutarate. So the products of this reaction, of course, is your alpha ketoglutarate, your carbon dioxide, as well as your NADH. So, tandaan nyo yung yield no reaction number 3, alpha ketoglutarate, carbon dioxide, and NADH. And remember that your reaction number 3 is the rate-limiting reaction because it has the rate-limiting enzyme, which is isocitrate dehydrogenase. Okay? Next. Let's go to the fourth step of the Krebs cycle. So the fourth step is also a similar reaction wherein there is oxidative decarboxylation. So, and the enzyme is also a dehydrogenase kasi it's also an oxidation reduction reaction. So you have alpha ketoglutarate dehydrogenase complex. So kaya siya naging complex kasi maraming required na coenzymes ng si alpha ketoglutarate, but we don't really need to go in depth with the alpha ketoglutarate on our level. So, what happens here? There is a decarboxylation reaction. So, sabi natin, pag decarboxylation, di ba, mayroong removal of a carbon dioxide or removal of a COO. So, pag tinanggal yan, anong papasok dyan? Ang papasok dyan is your OH. So, the decarboxylation occurs, forming CO2. And then, the OH will react with your alpha ketoglutarate in order to form your succinyl-CoA. Okay? So, itong H naman nito will combine with NAD plus in order to form your NADH. So, ganun yung nangyayari dito. So, pag tinanggal yung H dun sa CoH, it will combine with NAD to form NADH, and then the CoH will bind to the original alpha ketoglutarate in order to form succinyl-CoA. So, yun yung nangyayari dito sa reaction number 4. Next. So, now we have your succinyl-CoA, di ba? So, kanina, from citrate, naging isocitrate. Si isocitrate, naging, Si isocitrate, naging alpha ketoglutarate. And then your alpha ketoglutarate becomes succinyl-CoA. So, your succinyl-CoA now then becomes converted into succinate. So, succinyl-CoA to succinate. So, how does it happen? The enzyme involved is the succinyl-CoA synthetase. So, madali lang siya tandaan kasi before you because this your succinyl-CoA synthetase will synthesize succinate. So, pwede siyang reversible reaction. Okay? Kasi siya naging succinyl-CoA synthetase kasi pwede siyang gamitin to make your succinyl-CoA. So, what happens in this reaction? There is what you call the substrate level phosphorylation. Iba pa to dun sa oxidative phosphorylation which will happen in the mitochondria during the electron transport chain. This one is substrate level. So, you're working at a substrate or a specific molecule and you are adding a phosphate to that substrate. So, in order, you are getting a phosphate from the substrate and putting it into a phosphate manually. So, ganun yung nangyari dito sa substrate level phosphorylation. No? So, your succinyl-CoA, ito yung reaction na, di ba? Meron siyang CoH dulo. The CoH gets released and the phosphate binds to this portion of the molecule. Kaya siya nagiging succinyl phosphate. And then the phosphate from the succinyl phosphate gets added to the GDP molecule. So your GDP becomes a GTP, which is also a guanosine triphosphate. So ang equivalent energy, so it has equivalent energy to your adenosine triphosphate. So in this step, you actually produce your energy molecule, an energy containing molecule, your GTP. Okay? Ulitin natin, ang nangyari? Your succinyl-CoA, matatanggal yung co-wash niya, and the phosphate will enter. So, pagpasok ng phosphate na yon, that phosphate will be transferred to a GDP. Yung GDP, phosphorylate niya, magiging GTP. Kaya para na siyang molecule na similar to your ATP. So, tandaan, between your succinate, you have your GTP. Okay? Next, you have your oxidation of your succinate. So, you have your succinate na, di ba? Galing na tayo dun sa succinyl-CoA synthetase forming your succinate. Then, your succinate now will also become oxidized. So, sabi natin pa ulit-ulit, maraming oxidation reactions na nangyayari dito sa ating Krebs cycle. So, one of these is your step 6, which happens to your succinate. Your succinate becomes oxidized by your oxidizing agent, FAD. So, kanina puro NAD yung kausap natin, di ba? Ngayon naman, FAD siya. Sabi natin, FAD, when you add that, when it serves as an oxidizing agent, you form a double bond between single bonds ng iyong dalawang carbons. Di ba? So, when you remove the H's here, you form a double bond. And that configuration of the double bond here is trans. So, in order to form your fumarate. So, the enzyme responsible for that, or this reaction is your succinate dehydrogenase. And an important thing about your succinate dehydrogenase is that this enzyme is found in the inner mitochondrial membrane. So, sinabi natin that your citric acid cycle occurs in the mitochondrial matrix. Except for one enzyme, and this is that one enzyme na except. Si succinate dehydrogenase is located in the attached to the inner mitochondrial membrane. Okay? So, balikan natin kung ano nangyayari. 2 H atoms are removed from succinate in order to form a trans double bond. So, yung isang H nandito sa isang side, and then the other H is on this other side. Kaya siya naging trans double bond. And then the 2 H atoms will combine with your FAD in order to produce your FAD H2. Okay? And this FAD H2 can later on enter the electron transport chain. Okay? So, next. So, after step 6, we'll go to step 7. So, what happens in step 7? There is hydration. So, ibang reaction naman to. Hindi na siya type or oxidation. There is hydration of fumarate. So, hydration, nagdagdag ka ng water, you added water. When you add water, pinaghiwalay mo yung OH and H na, yung isang H, yung H nilagay mo dito, yung OH nilagay mo dito sa carbon na to. So, in order to form your malate. So, your water is added across the trans double bond to produce your malate. Okay? And the enzyme responsible for that is your fumarase. So, medyo madali lang siya tandaan kasi fumarate enzyme, you use fumarase in order to form your malate. Okay? So, now let's go to the last step of your Krebs cycle, which is step 8, wherein your malate is oxidized in order to regenerate your oxaloacetate. So, anong enzyme ang responsible for this one? It's malate dehydrogenase. So, pag oxidation reaction involved, syempre, yung enzyme natin is dehydrogenase. So, what happens here? Your malate, you oxidize it. So, sabi natin, pag inoxidize mo, you will reduce the H's and you will give it to something else. So, yung H na ito, dinagdag mo kay NAD, kaya si NAD ay na-reduce. Si NAD naging NADH plus H plus, and then this one becomes oxaloacetate. So, So malate reacts with NAD plus to produce NADH with H, and oxaloacetate is regenerated. So pag na-regenerate mo na si oxaloacetate, it can combine with another acetyl-CoA to start the cycle again. So di ba ito yung oxaloacetate mo? And then since you have an oxaloacetate already, again, that oxaloacetate can combine with acetyl-CoA in order to form and to start the cycle again. Okay? So next. So what are the features of your citric acid cycle as we mentioned? So the fuel of the cycle is your acetyl-CoA, as we mentioned, which is derived from catabolism of carbohydrates, fats, and proteins. As mentioned, 4 out of 8 reactions involve oxidation and reduction. When you form a C double bond O, you use NAD as an oxidizing agent. When you form a double bond between your carbons, you use FAD as the oxidizing agent. The two carbons enter the cycle as acetyl-CoA, and two carbons leave as CO2. Okay? So in the cycle, as we mentioned earlier, B vitamins are essential for proper functioning of the cycle. Pero ito naman, hindi ko naman, so, an important enzyme na medyo maraming requirements is your alpha-ketoglutarate dehydrogenase because it requires thymine and riboflavin. Meanwhile, na-mention na natin itong tatlong important nucleotides na to, no? We know that FAD requires FAD, we know that NAD, your FAD requires B2, your NAD requires B3, and your CoASH requires B5. Okay? Next. So here is the net overall summary of the reaction in the citric acid cycle if you sum all of the reactions up. So starting is your acetyl-CoA. You use 3 NADs, 2 FADs, 1 GDP, an inorganic phosphate, and 2 waters. So ang product niya is actually dalawang carbon dioxide, isang CoASH, tatlong NADH, isang FADH2, at isang GTP. So when we want to compute if you want to compute the total amount of ATPs yielded from one acetyl-CoA molecule, so ito yung magiging resulta niya, no? So from GTP, sabi natin, this is equivalent to an ATP. So that's a total of 1 ATP. Your NADH can enter the electron transfer chain and undergo oxidative phosphorylation in order to yield 2.5 ATPs per NADH. Your FADH2 can also enter the ETC and yield 1.5 ATPs per FADH2. So, ganito yung total niya. So, 2.5 times 3, that's 7.5. 1.5 times 1 is 1.5 plus 7.5 plus 1, you produce a total of 10 ATPs per 1 acetyl-CoA molecule. Okay? So, your citric acid cycle is actually controlled controlled cycle, no? So the rate of which the citric cycle operates is controlled by the body's need for ATP. Siyempre, kung hindi naman niya kailangan gumawa ng maraming ATP, the citric acid cycle will slow down. Pero the more the body needs energy, the more that the body will go through the cycle faster. So here are some of the control points during the citric acid cycle. So your citrate synthase, which is the first enzyme that we encounter, is actually inhibited by ATP. So pag marami ng ATP, na-detect ng katawan mo, marami na akong ATP, hindi ko na kailangan mag-citric acid cycle. So the cycle slows down. So it will only speed up again when there is already large amounts of ADP. Pag marami ka kasing ADP, ibig sabihin your ATP is being broken down. So you're utilizing your energy and your body needs energy. That's why when there are high amounts of ADP, your citrate synthase will become activated and your citric acid cycle goes faster. Next. Another enzyme that is part of the regulation of your citric acid cycle is your isocitrate dehydrogenase. So your isocitrate dehydrogenase is inhibited by large amounts of NADH. Kasi nga yung NADH, di ba, is produced by the citric acid cycle. So pag marami ka ng NADH, ibig sabihin marami ka ng product, marami ka ng energy. So if mababa lang yung NADH, then if mataas yung NADH, then the body will slow down the citric acid cycle. Your isocitrate dehydrogenase is also activated by ADP. Same rationale as earlier. Pag marami kang ADP, pag mababa yung pag mataas yung ADP mo, ibig sabihin your ATP is being consumed, no? So, ibig sabihin, mataas yung energy utilization. Thus, your isocitrate dehydrogenase, uh, uh, the activity of your isocycle dehydrogenase is enhanced in order to make the cycle go faster. Next, another control point is your alpha ketoglutarate dehydrogenase. So these are inhibited by succinyl-CoA and NADH. So ito yung mga products of the reaction. So pag mas maraming product, syempre may inhibit niya yung reaction itself kasi hindi niya na kailangan mag-undergo the reaction dahil marami ka nang nabuo. And when the levels of these two go down, doon lang magpo-proceed ulit yung citric acid cycle ng mabilis. Okay? So, here is a summary slide of the citric acid cycle, no? So, the starting material is your 2-carbon molecule, acetyl-CoA. It will combine with a 4-carbon molecule C, oxaloacetate, in order to form your citrate. Si citrate, meron siyang 3 carboxylic acid, hence it's known as hence your Krebs cycle is known as a tricarboxylic acid cycle. So your citrate will undergo so your citrate is formed from the condensation of your acetyl-CoA and oxaloacetate. And then your citrate will undergo isomerization in order to form your isocitrate. So after isocitrate is formed, your isocitrate, sorry, your isocitrate undergoes oxidation and decarboxylation. So oxidizing agent used is NAD in order to produce NADH. And then there is decarboxylation. So there is production of CO2. So here is, and you produce your alpha ketoglutarate. Kasi nagtanggal ka ng carboxyl, so nabuwasan ka ng isang COO. So natanggal ka ng isang carbon. So this one becomes a C5 carbon. C5 carbon molecule, no? So your alpha ketoglutarate is later undergoes oxidative decarboxylation to see your NADH also, as well as releasing a carbon dioxide molecule. So from C5, magiging C4 siya, yung ating succinyl-CoA. Your succinyl-CoA is then phosphorylated to form your succinate. So, and then the phosphate from the succinyl phosphate will be transferred to your GDP in order to form your GTP. And then, you yield your succinate, which is also C4. Kasi, nag-phosphate lang naman yung dinagdag mo, hindi ka naman nagdagdag or nagbawas ng carbon atom. Okay? And then, your succinate is then oxidized using FAD as an oxidizing agent to form a FAD-H2 as well as fumarate. So your fumarate is then later on hydrated to form your malate, and malate is later oxidized with NAD to form NADH and oxaloacetate. So when you have your oxaloacetate, again, it can combine with another molecule of acetyl-CoA and the cycle starts again. Okay, next. So, here are some mnemonics on how to remember the steps of your Krebs cycle. So, yung reactions kayo na bahala mag-memorize. Pero, madali lang naman siya kasi pag may idea ka ng mga products for the reaction, you'll have an idea. So, here is the mnemonic para mabilis siyang tandaan. So, citrate is a starting substrate for making oxaloacetate. Citrate is a starting substrate for making oxaloacetate. So, tandaan mo na the first reaction is a condensation reaction that produces citrate. Your citrate then undergoes isomerization to form isocitrate. And then you form alpha ketoglutarate. So, tandaan nyo, kung anong step na napoproduce yung mga NADH at yung mga carbon dioxide natin sa cycle, no? So in step 3, you produce NADH and CO2. In step 4, you also produce NADH and CO2. And then in step 5, tandaan nyo, when succinyl is converted to succinate, you produce GTP. Next, when you have succinate to fumarate, you form FAD-H2. And then, another NADH is produced in step 8. So, ito yung tatandaan nyo. Important to. And then, the order of reactions as well as the enzymes. And tandaan that the rate-limiting enzyme of your citric acid cycle is isocitrate dehydrogenase. Okay? So, tapos na tayo dito sa part ng citric acid cycle, no? So, If you have difficulties understand, you can go through it slowly and replay this video since this is a recording anyway. So when you digest, uh, for pyruvate, your pyruvate gets converted into your 2-carbon acetyl unit, which is attached to CoWash to form your acetyl-CoA. Your acetyl-CoA enters the citric acid cycle to produce 2 carbon dioxide, GTP, and NADH and PADH2. So, ang key product ng citric acid cycle actually is the uh, cit is the NADH and the PADH2. And these two actually enter yung tinatawag nating electron transport chain. Okay?